The session was on liquid biopsy, and one focus of the question definitely was the type of tests we have, tumor-derived test versus tumor-agnostic test. The second talk was on the clinical utility, and my talk focused on possible endpoints. Now, if you talk about the SURVIVE study, which is a surveillance study in early breast cancer survivors, in which we randomize patients between standard of care surveillance versus intensified surveillance using liquid biopsy, including ctDNA testing, then we really have to look for overall survival, because if we detect asymptomatic distant metastases earlier going through liquid biopsy, then of course we shorten disease-free survival and distance disease-free survival with the goal of gaining on overall survival. Now, the vast majority of studies beyond that are focusing on patients with true molecular relapse. Molecular relapse defined as patients with molecular disease, ctDNA in peripheral blood, derived from either tumor-informed or tumor-agnostic tests, and then patients without metastasis at that time point. Now, depending on the test, depending on the timing, there is a certain percentage of patients who already have asymptomatic distant metastatic disease at that point of time, and they, of course, get standard care of treatment. Those patients who do not have any overt distant metastases on imaging, they are candidates for interventional trials, which were discussed yesterday. And in these trials, we probably can use IDFS and distant disease-free survival, as in most early breast cancer trials, or ctDNA clearance. Since we have a parameter which is ctDNA positivity or molecular relapse, this is also an instrument for monitoring.